Hey guys, so today we're going to be talking about eukaryotic gene regulation. Now this is something that is much more complex in general than prokaryotic gene regulation. We're going to take a look at the differences, and by the end of this video you should be able to recognize some of the main differences between the two different types of gene regulation in these two different types of organisms. Now, first of all, we're taking a look at prokaryotic DNA and eukaryotic DNA. Remember that prokaryotic DNA is organized in operons, we already know that, and eukaryotic DNA is generally much more complex. We have introns, we have non-coding regions, and we're going to see a lot of different types of transcription factors as well, so we'll take a look at that in a second. So, eukaryotic DNA is going to be arranged in chromatin, which is going to make protein-DNA interactions difficult unless we get that chromatin unwound somehow. And transcription and translation occurs in two different places. So the DNA is going to be always in the nucleus. Prokaryotic cells don't have a nucleus, so they have more of a nucleoid where the genetic materials condense together sometimes, but they're not going to have a membrane-bound nucleus. So transcription, it takes place in the nucleus. Translation will take outside of the nucleus where the ribosomes are. Now, there's many different points of control as far as eukaryotic gene regulation can go. So remember that the expression of a given gene won't necessarily go through all these steps that we see in this diagram right here, but there's lots of different points within this entire process of protein synthesis where our genes can be regulated. So some of those are the chromatin packings or modification of the chromatin, assembling of transcription factors, so at the transcriptional level, RNA processing, which we've already learned about, and then regulation of mRNA, degradation, and control of translation. So these are some steps that we'll talk about today on when the RNA will be broken down or when certain mRNAs will be translated into actual proteins. And then, of course, protein processing and degradation. So the main lesson is that each stage of protein synthesis, there is a potential control point, so we can have our genes regulated at all of these steps or none of these steps, and all these steps can be sometimes sped up or slowed down depending on what's happening within the cell. So let's talk a little bit about how organized DNA is. Now, DNA is very organized in eukaryotic organisms. So we have the DNA wrapping around different special proteins called histones. And these are proteins that are responsible for the first level of DNA packing in chromatin. And so chromatin is a complex of DNA and protein, and it's found in the nucleus of our eukaryotic cells. And these histones will group together to condense and coil up, and they'll become a nucleosome. And then those nucleosomes will supercoil into what we can see as chromosomes. And those chromosomes will mostly be condensed in chromosomal form that we can see in the stages of cell division. So especially metaphase, that's going to be the most condensed these chromosomes or the chromatin will ever be. And we've seen pictures of those. We know what the chromosomes look like through our microscope when it is totally condensed. Now we have what's called euchromatin and heterochromatin, and we'll talk about what that means. And of course, this nucleosome here with a bunch of different histones will have little tails of DNA that will be exposed, which will help with transcription. So let's talk about euchromatin and heterochromatin. First, euchromatin. These are going to be, will be the more loosely organized type of chromatin. So we'll have these loose acetyl groups, and they're going to be like sort of those tails that stick out that we saw in the last slide. So this is our acetyl group right here. And the euchromatin is going to be, because it's more loosely packed, it's more accessible, it'll be the chromatin that is transcribed. So this is the DNA that is most likely going to go through transcription. So when we say histone acetylation, it's going to loosen the nucleosome structure and thereby enhance transcription. So we'll see more transcription with our euchromatin. Now our heterochromatin is going to be more densely compacted. We'll have methyl groups on the tails, or the methylation, and this is going to diminish the transcription of DNA. So this is our methyl group here. And so in general, the heterochromatin will not be transcribed. So methylation is going to diminish transcription or levels of transcription. All right, so this is another diagram of DNA organized in the cell, or the levels of organization of DNA. So this is our double helix that we know is the original structure, and then that double helix is wrapped around complexes of histones to create nucleosomes, and then those nucleosomes will supercoil, and when we get this looped chromatin here, it can totally condense into our chromosomes that we can see in cell division. Now in here in this image, it may be a little difficult for you to see, the heterochromatin, so the stuff that won't be transcribed is the darker stained part, and then the lighter stained is going to be our euchromatin here. And then this is the nucleolus, the very center of the nucleus. And again, we have another nucleosome right here. All right, so moving forward. Let's talk about transcription factors. Now, these are special proteins that mediate RNA polymerase, and the control of transcription factor availability is a huge way multicellular organisms can differentiate. So this is called differential gene expression, and this is so different cells can have different functions and do different things in the organism. Now, there's a lot of different types of transcription factors and activities, but there's specific sites known as enhancers are going to bind with our transcription factors and, with our promoters, and these transcription factors can interact with the promoter, and sometimes this can be like thousands of nucleotides downstream, and then we'll have this bending of DNA. So what happens is activator proteins are going to bind to the enhancer sequence in the DNA to help position the initiation complex, and then DNA bending, so you'll see sort of this folding over, is going to bring the bound activators close to the promoter, and then other transcription factors in RNA polymerase are nearby, and then we'll have these protein binding domains on the activators attached to certain transcription factors to help them form an active transcription initiation complex on the promoter. So just know that the transcription factors can either increase or decrease our rate of transcription depending on what they do and their availability. And of course, RNA polymerase will initiate transcription as always. However, it's due to the dependence of these transcription factors and how they're interacting with the promoters on these certain parts of DNA. Okay, so we've already talked about alternative splicing. One thing that's special about eukaryotic DNA is that we have introns and extrons. Introns and exons. And these introns are our intervening regions, and exons are expressed regions. But due to alternative splicing, we can get multiple types of proteins from the same stretch of DNA, depending on which exons are actually kept and which ones are cut out. So we talked about this already in our RNA details video, so please check that one out if you'd like to learn more about it. Now, remember, there is mRNA processing that happens as well. And this is primarily due to the additions of the 5' cap and the poly A tail and whether or not those are there. And these are going to help with the stability of our mRNA and whether or not they'll be degraded by certain different proteins before it reaches the transcriptional level. So we also already talked about this in our RNA video, so feel free to go back and check that one out as well. Now, microRNA, or sometimes we'll see like miRNA, is going to inhibit the translation of specific mRNAs, causing premature degradation. So whether or not the microRNA takes apart these mRNAs is going to be another level of regulation in eukaryotic DNA. Our diagram right here. And then, of course, our proteins can be targeted for degradation as well. So proteins can be tagged and broken down at any point in their processing. So we'll have this thing called ubiquitin. It's going to direct the targeted protein to a proteasome. And the proteasome is a complex structure where proteins are digested by several power proteases. And proteases, of course, are enzymes that will break down proteins. Remember, enzymes are proteins anyway, so these are special proteins designed to break down other proteins, and then we'll have our protein fragments, which are just peptides within the cell if the protein isn't set to function. All right, so let's talk about prokaryotic versus eukaryotic gene regulation. Now, remember, prokaryotic cells are organized in operons, and eukaryotic cells, the genes are generally spread out. For prokaryotic cells, generally we will have one promoter for multiple genes. But in eukaryotic cells, we will have what biologists think is one promoter for one gene. So one promoter goes along with one gene. Now, in prokaryotic cells, we have no introns, but eukaryotic cells, we have many different types of introns and many introns within one section of DNA. In prokaryotic cells, as far as organization goes, some can coil, some have protein, but it's very different from our highly organized DNA in eukaryotic cells with histones, and it's greatly condensed as chromatin. In prokaryotic cells, we have no nucleus, so transcription and translation don't wait. In eukaryotic cells, we have additional control mechanisms because transcription and translation are going to take place in different locations. And so this compartmentalization is going to allow us to have more points of control in the eukaryotic regulation process as far as the regulation of gene expression goes. Okay, so that was a lot. It was fast, but please make sure you have in mind a few different points of regulation for eukaryotic gene regulation, and know the differences between prokaryotic and eukaryotic gene regulation as well. All right, so if you need to take a little bit longer look at this slide, here it is. I can't get my face out of the way, sorry. And we'll see you later. Bye.